In this video, we're going to be looking at the evidence for the obligate coupling of oxidation in the electron transport chain and ATP synthesis. So what does that mean? What that means is that the process of oxidizing NADH and succinate here within the electron transport chain, or in other words, the consumption of oxygen, is coupled to the production of ATP. And the reason for that is because it is this consumption of oxygen, the oxidation of succinate and NADH, that provides the energy necessary to pump protons from the matrix into the intermembrane space. And then it is that proton gradient that is used by ATP synthase to make ATP. So those two processes are coupled. And let's take a look at the experiments that can show us how they are coupled and then show us what we can do to uncouple them. And all of this, the reason for these experiments is simply to demonstrate that yes, indeed, the proton gradient from the electron transport chain is what is used to make ATP via ATP synthase. So what the experiments involve is taking isolated mitochondria that have been taken from cells and incubating them in a solution. In that surrounding solution, we can add stuff to, we can take away from, and as we do that, we're constantly measuring the consumption of oxygen. Okay, that's going to be our marker of how much oxidation is occurring within the activity of the electron transport chain. And we will be measuring the amount of ATP that's produced. That is our measure of the activity of ATP synthase. So the data will look like this. In black is the amount of oxygen that is consumed. And so the higher up we go, the more we have oxygen we've consumed. In red, we have ATP synthase. And so again, when we go up, that means we are synthesizing ATP. Now notice, here we start off with nothing added to the isolated mitochondria, and so we don't get any consumption of oxygen and we don't get any ATP synthase. Then we add to it succinate. Now that is our source of electrons that is going to be run through the electron transport chain, and that is going to allow protons to be pumped. But notice we don't get any production of ATP until we get ADP and inorganic phosphate added. So makes sense, right? If you don't have any ADP or inorganic phosphate, you're not going to get any ATP made. But what also is interesting, though, is that even though we have this succinate here, a source of electrons for the electron transport chain, we don't get any consumption of oxygen. It's not until we add ADP or inorganic phosphate. So it's these two processes are coupled together. If one is not rising, the other is not going to rise. Let's take a look at why we don't get the consumption of oxygen until ADP and inorganic phosphate is added. So here we have the electron transport chain, and here's a reminder that complex 3 and complex 4 are enzymes, and they are catalyzing reactions that have reactants and they have products. And when we visualize it like this, like an image, then it's hard to imagine it like a reaction, but it's important that we think about it as a reaction. Because here's the problem. We're adding succinate. Now that succinate is going to reduce QH2, and that QH2 is going to be fuel for complex 3, and then the cytochrome C is going to be fuel for complex 4, and that fuel is going to be used to pump protons against their concentration gradient. Now, how far can the cell go? Right? How many protons can it move into the intermembrane space? Can it go forever? In normal situations, it won't really matter because those protons will be pumped out and then they'll go back through ATP synthase back into the matrix. And so the concentration, although will always be higher than that in the matrix, will never be super high because they'll always be flowing down their concentration gradient here through ATP synthase. However, if ATP synthase is not bound to ADP and inorganic phosphate, it does not allow those protons to move down their concentration gradient. So we're at a situation here, because we haven't added ADP, we haven't added inorganic phosphate, those protons are just going to build up in the intermembrane space. Well, here's the thing. Those protons in the intermembrane space are products of the reactions catalyzed by complex 3 and complex 4. And like any product, if it increases in concentration too much, it's going to take this reaction that is normally exergonic and it's going to reach an equilibrium. And what that means is that no matter how much succinate you add here, the concentration of the protons here is going to stay the same. Okay, we cannot drive that reaction enough that there is a maximum concentration of protons that can exist within the intermembrane space until reactions reach equilibrium. And at that point, we can no longer oxidize cytochrome C with oxygen, and therefore we can no longer consume oxygen. Therefore, we can no longer oxidize QH2, and therefore we can no longer oxidize succinate. Which means that if ATP synthase is shut down, for example, by not giving it ADP, those protons can't get through, they build up, and then even though we have a source of electrons, succinate, even the electron transport chain shuts down. And that's what we're seeing here. Even though we've added succinate, we still don't get oxygen consumption until we add ADP and inorganic phosphate. And now we have both the production of ATP coupled along with the consumption of oxygen. Fantastic. But now let's see what happens. Again, we're messing around with the system to support the idea that these two processes are coupled. They are dependent upon one another. So the next step is we add cyanide to the system. And now what cyanide does is it blocks complex 4. And so, okay, you block complex 4. Complex 4 catalyzes the reduction of oxygen to water and the oxidation of cytochrome C. So yeah, if you block that, of course, you don't get any O2 consumption. But take a look at this. We haven't taken away ADP, inorganic phosphate. That is still there. We haven't taken away any of that, and yet we don't get any ATP made. So why is that the case? Well, if you block cytochrome, if you block this complex 4 with cyanide, then that means we can no longer oxidize cytochrome C, which means we can no longer oxidize QH2, which means we can no longer oxidize succinate. So all of this stops, which means we're no longer pumping protons into the intermembrane space, which means there is no more proton motive force that can be used by ATP synthase to make ATP. So, again, we have demonstrated that they're coupled in that if we shut down the electron transport chain by blocking complex 4 with cyanide, we also block ATP synthase and the production of ATP. So let's look if we could do the opposite. Let's see what happens if we block ATP synthase. Will that stop the electron transport chain and the consumption of oxygen? Let's take a look. So we do the same experiment, and yes, add succinate, nothing. Add ADP and inorganic phosphate, yes. Now we have both of them working. We've got the consumption of oxygen in the electron transport chain, which is giving us our proton gradient, and therefore that proton gradient is being used by ATP synthase to make ATP, so we're going up, 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 up. But at this time point here, when we add ventricidin and oligomycin, these are both drugs that block ATP synthase. So yes, if you block ATP synthase, sure enough, you won't make ATP, and therefore that levels out. But take a look at this. The consumption of oxygen also levels out. They are coupled. If you block one, you block the other. Let's see why that happens. So this is very much like the situation in which we don't have ADP and inorganic phosphate. In this case, we do, but we've added this drug which blocks the entire ATP synthase enzyme. What that means is protons can no longer move through ATP synthase. They can't go down their concentration gradient. And that means that all of these reactions are going to reach an equilibrium such that no more protons are going to be pumped against their concentration gradient, and no more oxygen is therefore going to be consumed. Right? Because there's only so much protons that can be pumped here until these reactions will no longer continue to move in the forward direction, and we're no longer consuming oxygen. And so that's the reason why when you shut down ATP synthase, not only do you lose ATP production, but also you lose O2 consumption. All right, now let's think about something that can uncouple these reactions. And by that, what I mean is there a way that we can consume oxygen without making ATP? Can you think of a way in which we can get the electron transport chain going again after ATP synthase has been blocked? There is a way. You might be able to predict it. The answer are molecules called uncouplers. And there's several different types of uncouplers, but what they have in common is they are alternative ways for protons to move through the inner membrane of the mitochondria back into the matrix from the intermembrane space, bypassing that blocked ATP synthase. So for example, this one here, DNP, it holds a proton here. It's nonpolar, so it slides right through the membrane into the matrix and then lets that proton leave. Or we have FCCP, same thing. It's nonpolar and it holds onto a proton, so it goes straight through the membrane, bypassing ATP synthase all the way back into the matrix, and then it releases that proton again. So what these uncouplers do is they allow another way for protons to move down their concentration gradient into the matrix, bypassing ATP synthase. So let's take a look at how that data would look when we did the experiment. Okay, we start way off here to the left. We add succinate, but we still don't get any consumption of oxygen or the production of ATP. We go, bang. Now we add our ADP and inorganic phosphate. We open up that ATP synthase for those protons to move through. That allows us to consume oxygen, right? Allows us to keep pumping protons into that intermembrane space. We are, of course, making ATP by using that concentration gradient via ATP synthase. And now time goes. They're both and then we shut this down. Okay, we shut ATP synthesis down, or ATP synthase down. Those protons can no longer go through it. They build up in the intermembrane space. So not only do we not make ATP, but we can no longer consume oxygen. And we go, and then suddenly now we add one of those uncouplers. One of those uncouplers that's going to allow protons to move down their concentration gradient and not dependent on ATP synthase. They're just going to go right through that right through that membrane. What that means now is the concentration of protons in the intermembrane space will go down. which will relieve the pressure and allow these reactions to run again, allowing us to consume oxygen again. And that's what's happening. But notice it's happening in the absence of ATP production. So we have, via these uncoupling molecules, we have uncoupled ATP synthesis from the consumption of oxygen in the electron transport chain. And all of the purpose of all of these experiments is to just find different ways to demonstrate that, yes, the theory of this chemiosmosis, of the proton motive force being the driving force for making ATP, are all supported by these various experiments.